Mentioned, my name is Jade Arneson. I'm a recent UW Green Bay master's graduate. So I graduated from their environmental science and policy program back in September, so fairly recently. I also work for Glacierland Resource Conservation and Development, helping private landowners with various natural resource concerns that they may have on their properties, and working with them through some of the available cost sharing opportunities with NRCS programs. Prior to this job and prior to graduate school, I worked for the Fish and Wildlife Service for a few details within their fisheries department. I also worked for UW-Madison within their Forest and Wildlife Ecology department. And I worked for the Nature Conservancy up in Sturgeon Bay doing some habitat restoration work. And even prior to that, I'm happy to say that I got started with some of my very first conservation experience professionally at Woodland Dunes. So it's been neat to kind of come full circle and join Woodland Dunes tonight for this presentation. So very thankful to them again for inviting me. And what I'm going to be talking about tonight is about my graduate thesis work, which was on restoring wild rice in Green Bay coastal wetlands. And it looks like I'm having an issue advancing slides. There we go. Can everybody see that? Okay, so wild rice is an aquatic grass in the genus Zizania. There are 3 species of wild rice in North America, and I'll start on the far left. They include Zizania aquatica, Zizania palustris, which is actually the species that I worked with in my thesis work, and then also Zizania texana. And so these 3 species have been contracting in range throughout the past years, largely due to climate change, but there are a variety of other confounding factors that are influencing that. So wild rice is an annual plant, and what I mean by that is it's dependent on producing seed every year in order to sustain its population. And how much seed it produces in a given year can vary. Generally, there's a 4-year cycle that wild rice populations can go through, with 1 year being a boom year, 1 year a bust year, and then 2 years being kind of mediocre. And that's all dependent upon the environmental conditions that are present at the site where wild rice is growing. So the life cycle of wild rice, to me, was easily one of the most interesting things when I started this project. So I think when most people think about wild rice, the image that comes to mind is either the grain that's ready for consumption, or it's the emergent plant within the marsh. But I don't think many people are aware that wild rice actually starts out in a submergent phase in late spring, early summer. So that's pictured here at the top. And it'll then progress counterclockwise to floating leaf stage. So if you look out across a water body, what you're going to see in about early to midsummer is just this mat of like light green ribbon-like leaves that are floating on the surface. And then it'll progress to its emergent stage that people are most familiar with in about mid to late summer. And it'll progress into flowering, where it's then wind pollinated, and it'll produce seed that eventually matures and falls off and settles into the substrate, where it will then wait out over the winter and hopefully germinate come spring when conditions are favorable. And this complex phenology of wild rice makes wild rice susceptible to different chemical, biological, and physical factors throughout each of its growth stages. Some of those factors are pictured here. There's very many that have been documented in the literature. One of the most well-known environmental factors is water depth. So of course, in the Great Lakes region, the past couple of years, we've been seeing an increase in water depths, right? Nobody's a stranger to that, especially within Green Bay. Water depth is known to especially affect wild rice when it's in that submergent and floating leaf stage. So when wild rice is submerged, if water levels are increasing, the plants are constantly fighting to break the water surface and to get enough light in order to do so. And that can actually lead to really weak, attenuated growth. And in some circumstances can lead to the plants not actually emerging and producing that seed head like we need them to in order to be self-sustaining. In floating leaf stage, if water levels increase too quickly, the plants can actually become uprooted just from the buoyancy of their own leaves. Another factor is just the increase in frequency of storm events and the severity of those storm events. So wind and wave action can, of course, uproot these plants, especially when they're in that submerged and floating leaf stage and they don't have that root structure established yet. And this is confounded by the type of sediment that the plants may be growing in. So if the substrate is too hard or too soft, that may lead to poor root anchorage and increased susceptibility of plant mortality. Some other factors are related to biota, so things like Canada geese or common carp. Canada geese are well-known grazers on wild rice, especially in their brood rearing stages. They can come through and just mow down an entire bed of wild rice. And this is especially detrimental when wild rice is in those earlier growth stages and at wild rice beds that maybe are actively being restored and don't quite have that density there yet to protect them from a fair amount of grazing. And common carp, people are very familiar with carp. They're going to be rooting around in the sediment, stirring that sediment up, making the water more turbid, attenuating light, and they're also going to be uprooting plants. And also in the literature, you'll find some research on competition with other aquatic plants. So things like yellow pond lilies that may shade out that early growth wild rice. On the flip side though, there's actually been some facilitative roles from other aquatic plants. There's research that showed that wild rice that was grown in association with robin's pondweed actually fared better than wild rice that wasn't. And they believe that was due mainly from nutrients, specifically phosphorus. So there's a variety of factors at play here that are well known in the literature, some of which are confounding factors. So someone had joined on earlier and they asked, well, why is wild rice important, right? Why do we care? Why am I tuning in today? Why are we trying to restore wild rice to the Green Bay system? So there's a variety of reasons that make wild rice important. Perhaps one of those biggest reasons is that there's a cultural and spiritual connection for First Nations people within the Great Lakes region. So Lake Superior Ojibwe were actually told by their creator in their origin stories to go to the place where food grows on the water. And that food was wild rice or manumin. And not only is it a food resource for people, but it's also a food resource for wildlife like waterfowl and it provides really great nesting cover for marsh birds as well as larval fish habitat. And in addition to that, people love to go up north in the fall and participate in wild rice harvesting. So there's recreational benefits associated with it, and along with that, economic benefits. So a lot of reasons to really care about preserving this plant and restoring this plant to the Green Bay system. So within Green Bay, there are quite a few historical anecdotes that talk about the distribution of wild rice within Green Bay and within its tributaries. And one of those anecdotes is from Jenks in 1901. And he stated, Green Bay, from above the mouth of the Menominee River, southward to the Bayhead, has been fringed with the plant from the earliest historical times, and today there are thousands of acres of wild rice in the shallows of its waters. There are some other accounts from Father Marquette in the late 1600s during his journey up the Fox River that talk about actually having to part the wild rice in order to maneuver the canoes, and just having throngs of waterfowl come up off of the Fox River from these wild rice beds that were present. And of course, in addition to that, the Menominee Indian tribe of Wisconsin and the Ho-Chunk Nation, both known as wild rice gatherers, were present within the region at the time of European settlement. So there's a lot of information out there that kind of alludes to the fact that wild rice had a fairly big presence on the bay historically. There are some herbarium records at UW Green Bay that show that some of the most recent records of wild rice were in the mid to late 1900s. And those records were from Peters Marsh, just on the other side, the leeward side of the Cat Island chain where my cursor is, and then in Duck Creek, just to the west of the Fox River. Today, though, however, that distribution has contracted significantly, with wild rice even disappearing from some regions of Green Bay, specifically the southwestern Lower Bay, which is pictured here. Practitioners within the region believe that wild rice disappeared from Lower Green Bay about the mid-20th century. The exact reasons why are not well known. It's likely a combination of factors, but not having that distribution and that abundance anymore has certainly left an unfilled ecological niche within the Green Bay system. So I feel like most folks in this presentation today are very familiar with Green Bay, likely have recreated on Green Bay, or maybe lived within the area at some point or do currently. So I'm going to imagine that folks are no stranger to some of the ecological stressors that Green Bay experienced, some of which likely led to the decline in the distribution and abundance of wild rice historically, right? So, we had the lumber era, we had settlement along the Green Bay West Shore, the industrial era on the Fox River with PCBs entering the water. Agriculture today is very prevalent within the watershed, so a lot of sediment and nutrient loading is occurring and contributing to eutrophication of Green Bay, and of course things like invasive species as well. So there's a lot of stressors acting on the system that may very well have influenced the decline in wild rice. Despite that, though, Green Bay really is a model ecosystem for freshwater restoration worldwide. There's been a lot of time and a lot of resolve dedicated to making conditions on Green Bay more favorable. Some of the recent efforts have been the restoration of aquatic vegetation. like wild rice. And that is what I will be talking a bit more about, and that is what was the focus of my research. So here is a map of the geography that I worked in for my graduate research. The triangles indicate 6 priority coastal wetlands where we seeded wild rice. The first wetland is Seagull Bar up in Marinette. The second is Lower Peshtigo River near the Peshtigo Harbor. The third is Oconto Marsh near the Oconto Breakwater. The fourth is Dead Horse Bay, or Longtail Point is another way it's known as, in Suamico. The fifth was Duck Creek Delta in Lower Green Bay on the leeward side of Cat Island. And then lastly, Point Sable on the east shore of Green Bay in the town of Scott. So these 6 wetlands were chosen because they were initially indicated in a Green Bay restoration project proposal. They also had ample public lands access, and there was already a presence from multiple agencies working there on various habitat restoration projects. So there were 2 main objectives for my thesis project. The first was to delineate regions of similar environmental conditions among wild rice restoration sites within Green Bay, and to try to relate those conditions back to wild rice cover. And the second was to improve understanding of the seasonal effects of various abiotic and biotic environmental factors on wild rice cover among our restoration sites. So in order to address these 2 objectives, we hand seeded wild rice at 18 discrete restoration sites in 2017 and 19 discrete restoration sites in 2018. And we seeded at a rate of 50 pounds per acre in the fall, specifically in November, right before ice up. The reason being that wild rice is required to have a cold hardening phase in order to break dormancy in the spring and germinate. And what I mean by a cold hardening phase is basically there needs to be this extended period of time where the seed is exposed to cold temperatures, so that come spring, when things start warming up, it can break that dormancy and germinate. And the source of our seed was Northern Lakes in Minnesota. What we then did come the growing season in 2018 and 2019 was go out to each discrete restoration site bi-weekly. We did this by boat and by canoe from about May through September to check up on our restoration sites to see how they were responding. And we measured a variety of factors, one of which, of course, was wild rice cover. And others included parameters such as water clarity, water depth, substrate characteristics. So how hard or how soft is the substrate? Is it sandy? Is it mucky? We also looked at non-restored aquatic vegetation cover, so floating aquatics, submerged aquatics, and other emergent aquatic plants, and measuring that cover. We also measured dissolved oxygen, water temperature, flow, and also made notes as to whether or not we noticed grazing within the wild rice beds. We also did put out a couple of camera traps at several of our restoration sites just to get a feel for which species may have been utilizing wild rice. So when it came time to statistical analyses for my thesis, The restoration site was the unit of analysis. So all that data that was taken at the restoration site biweekly from May through September, that was the unit of analysis. And what we did was we then reduced the data down to both an annual average by restoration site and a seasonal average by restoration site. And there were 3 seasons that I chose to parse my data down into. They included spring, early summer, so from about May to June, summer, so June to July, and then late summer, fall, August through September. And those seasons were intentionally chosen to reflect the discrete growth stages of wild rice, because one of our objectives was to see if environmental factors were influencing discrete growth stages differently or not within Green Bay. So in order to address our 2 objectives, we used 2 methods. The first was principal components analysis, which is a form of ordination. It's also known as PCA. And basically, it's a way of taking a really big data set like ours that has multiple variables that may be correlated and kind of parsing it down, making it visually easier to see and interpret, and kind of narrowing it down. And we used PCA in order to visualize how environmental conditions that we measured were relating back to wild rice cover. The second method was regression, specifically principal components regression and linear regression, to see with what % accuracy environmental factors were predicting wild rice cover. All right, so I'm going to talk about that first objective. So trying to delineate regions of similar environmental conditions among our restoration sites. So pictured here is a principal components analysis figure. This is one of many iterations that we ran. And again, those were looked at both years as well as for the seasons within the years. So there were many of these that I looked at, and they can be a little overwhelming, a little messy. So really all we need to pay attention to are the colored points that are plotted. So those represent the individual restoration sites. And again, there's 19 of them. And I have them color coded by wetland. The second thing to pay attention to are the vectors or the arrows. So those represent the different environmental parameters that we measured. And their magnitude and their direction is going to be important. So the greater the magnitude that they are, the greater their relative importance. And then also depending on the direction, so if they're moving in the same direction, they're positively related to one another. And if they're in opposing directions, they're negatively related to one another. So what we can start to do is we can start to see how wetlands are falling out among an environmental gradient, how the restoration sites we seeded within those wetlands are falling out among an environmental gradient, and how those environmental parameters we measured are relating back to wild rice cover. So right now in this figure, you can see that seagull bar in the light green here in the lower left quadrant. The 3 restoration sites there are really quite similar. They're clustered together quite closely. And the vector that seems to be associated with them is water clarity. So there's a really strong water clarity component there. As opposed to the restoration sites directly opposite that up in the top right quadrant. So these are going to be restoration sites that had lower water clarity. And you can see what some of them are. So we've got like Peshtigo Dyer Slough, Duck Creek Delta, Kenyer's restoration site. And you can also start to see that certain wetlands have restoration sites within them. They're actually quite different from each other. So if we look at Lower Peshtigo River in the orange, you can see that the channel restoration site and the lagoon restoration site are quite similar to one another and very similar to restoration sites in Seagull Bar, but they're very different from their counterpart within the same wetland. So what we learned from this is that, yes, environmental conditions really are quite different across the restoration sites where we seeded wild rice. And not only are they different by restoration site, they're also different by wetland. The next thing that I did was I overlaid wild rice cover as a grouping variable. So same figure, but instead the restoration sites are now grouped by wild rice cover, with the darker symbology relating back to greater wild rice cover. And again, we start to see some patterns. So your eye really wants to go again to this lower left quadrant where the symbology is darker, which again refers to greater wild rice cover. So you can start to learn which sites had greater wild rice cover. And then we can also start to look at the vectors that are associated with those sites with greater cover. So again, we're seeing water clarity seems to be playing a role. Emergent vegetation cover seems to be playing a role. If we look opposite these sites up in this quadrant, you start to learn a bit about, what may not be related to sites with greater wild rice cover. So for example, having softer sediment and having muckier sediment was a characteristic not associated with sites with greater wild rice restoration cover. So again, ran quite a few of these, looked at many PCA figures, and really was able to kind of start to learn what the overarching trend was in terms of relationships between environmental parameters that we measured and the wild rice cover at our restoration sites. And again, it really appeared to be that water clarity, some measure of sediment, and then non-restored aquatic vegetation was really driving these differences among wild rice cover at our sites. So the second objective was to then, using what we learned from the PCAs about which environmental parameters may be influencing wild rice cover, was to then take those parameters and use linear regression to find which parameters were significant and how well they could predict wild rice cover. So the four parameters that we ended up regressing against wild rice cover were water clarity, sediment composition, floating vegetation cover, and submerged aquatic vegetation cover. What we learned was that those four environmental factors had very little seasonality. So again, we had those three seasons: spring, early summer, summer, and late summer, fall, which again corresponded with the discrete wild rice growth stages. And what we saw was that there was very little seasonality. So the same environmental factors were showing up again and again across seasons to be important. Furthermore, the ability of seasonal environmental conditions to predict wild rice cover was inferior to, or maybe only marginally better than, the annual environmental conditions, except for late summer fall. So late summer fall was actually able to account for 59% of the variability in wild rice cover in 2018 and 70% in 2019. So that's a lot of variability that conditions in late summer fall were accounting for in wild rice cover. So that said something to us. So we looked a little bit further at the late summer fall conditions explicitly to determine which environmental predictors or factors were best predicting wild rice cover. And they included water clarity, sediment composition, and submerged aquatic vegetation cover. So, in other words, restoration sites that had greater wild rice cover had relatively greater water clarity. They had sediment that was hard and sandy, and they at least had a presence of submerged aquatic vegetation, if not a notable density of submerged aquatic vegetation. So, if we keep in mind these three factors, water clarity, submerged aquatic vegetation, and sediment composition, and we take a look at this figure, which is plotting wild rice cover by year and by restoration site, start to see that there must be something else at play that was relating back to these differences in rice cover between years that we observed. So, on average, there was a 15% decrease in wild rice cover from 2018, which is the very light gray color, to 2019, which is the darker gray color. So, you see this trend across almost all of the restoration sites, except for, I believe, one restoration site. And this, we believe, was largely due to the increase in water depths that we observed. So there was, on average, a 28 centimeter increase in water depth from 2018 to 2019 among our restoration sites. So that's almost a foot that water levels increased between years. The cool thing to note, though, is that despite that increase in water depths, restoration sites that, again, had that greater water clarity, had the harder, sandier substrates, and had relatively greater submerged aquatic vegetation, still performed better than the restoration sites that didn't have that. So if we bring this all together and hit on the most important things that I learned from my research and tie everything back to sort of management and policy implications, we did observe differences in environmental condition, not only among wetlands but also among restoration sites within wetlands, and that was what we expected. But in addition to that, late summer, fall environmental conditions best characterize similar restoration sites. Furthermore, differences in wild rice cover occurred among restoration sites and between years, which really stresses that need for site-specific analyses prior to seeding wild rice at a restoration site. And also kind of, keeping in mind the adaptive management strategy, right? So being able to adapt to changing conditions, especially within a system like Green Bay. Again, water clarity, submerged aquatic vegetation cover, and sediment composition. We learned are three metrics that may serve as very valuable indicators of a successful wild rice restoration site. And that furthermore, success can be reasonably predicted by sampling for these metrics in late summer, early fall within Green Bay. And another cool thing about being out there during that time of the year, sampling these metrics to determine where's a good site to seed wild rice, where is there not a good site, is that wild rice is emerged at this time, right? So it's really easy to identify, it's really easy to observe. If someone in the future wanted to also count stems or take seed production measurements, things like that, they would also be able to do that in late summer, fall with the plants emerged. So the hope is that what I learned from my research can be used by agencies like Ducks Unlimited, who really have been leading the charge with aquatic vegetation restoration in Green Bay, especially wild rice, so that they and other universities and agencies can take what we learned and go out across a broader range of wetlands in Green Bay and sample for the 3 metrics to see quite reasonably well where a site would do best for wild rice. So I can't end this presentation without acknowledging the many people that helped make this project a success. This would not have been possible without the manpower that everybody brought and the resources that everybody brought. So I would like to thank Ducks Unlimited, U.S. Fish and Wildlife Service, Wisconsin DNR, O'Connell Sportsman's Club, Mole Lake Band of Lake Superior Chippewa, UW Green Bay, and UW Extension for all of their help, and hopefully I'm not leaving somebody out. And then also to our funders, so the National Fish and Wildlife Foundation, U.S. Fish and Wildlife Service, the Fox River Trustee Council, and then also UW Green Bay. So with that, that concludes my presentation. I'm happy to take questions now. And also if anybody would like to get a hold of me at some point with further questions, or if folks would like a copy of my digital thesis for their own references, just feel free to email me. I have both my university and government email address listed here. So thank you very much, and I'll take questions.